in our lab, we most commonly use a UV coupled to the mass spec. So that's looking at how different wavelengths of UV light is being absorbed, which obviously we can't see that. But it is still based on the basic principles of light hits a sample, light gets refracted, and then you can compare how is that light refracted. And that gives us the visual information. So it's very
And
similar.
yeah. And like Erica was saying, a tart cherry is red because of its anthocyanin content. The redder it is, you could assume the more anthocyanins are present, and that's really based on the color. And I think a really simple one is any plant that's green, you could very confidently say contains chlorophyll because the green color is coming from chlorophyll. Plants need chlorophyll. We see chlorophyll as green. So that's a really simple
Definitely.
way of doing it. And then analytical tools just take that to the next level and can interpret things that are otherwise invisible to us.
So
Definitely.
speaking of green, let's talk a little bit more about basil. I'm also really excited that you mentioned that you grew 30 different varieties of basil.
Hmm.
Um, and just a short little anecdote, we bought some herbs last year from like a specific herb grower. And I had never encountered so many different varieties of basil there. There
Hmm.
were a bunch of really cool, really strange, brand new varieties. I'd never experienced some with like sort of exotic fruit scents as well.
Wow.
And we tasted them too. We would just like take a little leaf and we would taste them. And some of them tasted like the name or like the sort of exotic fruit that it might taste like. But some of them just tasted like plain basil. But I had no
Right.
idea that there were just so many different kinds. And this was the case for basil, but also a lot of the other herbs as well. Um, so
Hmm.
this what you said just really reminded me of that experience. And how many different varieties of basil did you say that there are?
There's over 60 species, and
Species.
then within each species, there's different cultivars. So cultivars are things that humans have kind of changed the chemical profile of. So, sweet basil, there's purple basil, and then there's your common sweet basil. The cultivar is plant breeders choosing which traits they want to optimize and cultivating it to be different. But then there's different natural varieties. So, say the same species of basil grew in two different environments, and to survive in those different environments, developed different chemical features. That would be a natural variety. So, because there's so many species of basil growing in so many different parts of the world, there's a lot of different natural varieties, but it's also something we love to eat. So, there's a lot of cultivars as well. And that just makes the taxonomy and the variation very, very complex. And it's like that with most herbs.
Especially herbs that are really important to us or
Definitely.
financially important. I think, again, to bring up cannabis, that's a really interesting species because within that species, you probably have tens of thousands of cultivars at this point because it's been financially lucrative to breed the plant to
Hmm.
have different expressions, but it's all the same plant. And if you look at the chemistry, it's wildly different. If you smell it, it's wildly different. If you consume it, the effects are wildly different, but it's all the same species. And there's just hundreds, thousands of different varieties, very
Hmm.
distinct. And I think
And that
also,
makes
go ahead,
well, it
Evan.
makes ID hard for
Yeah.
cannabis, pretty straightforward because those tools they are very well funded in the ability
Yeah.
to develop tools. But for things like smaller plants that, sustanch or something that only a few companies are selling and there's only a few sources of it, that makes it harder to identify because of that type of variation. And how do we know? Is it the same species with just broad variation, or are they actually different species?
Yeah. And then of course the issue of botanical reference materials comes up because at the end of the day, you do oftentimes have to trust someone to send you a botanical reference material and you have to
Hmm.
get it verified. But how many botanists in the world can distinguish between Cistanche tubulosa and Cistanche deserticola? I'm actually not sure how different those plants are, and I wouldn't be surprised if 99.99% of botanists on the world have never even heard of Cistanche.
exactly. Um, yeah, I have a PhD in plant biology and I hadn't heard of half of the plants we talk about. So it's a cool thing to learn about, but definitely a little overwhelming.
Yeah.
Yeah, for sure.
Which
And
is a compliment for us too, because it means we've been trying to specialize in novel niche plants that not everyone is dealing in. Which, yeah, it's become quite overwhelming for I think all of us. But we're really on a mission to discover all of the different types of plants that are out there. And it would also be really interesting to get into different cultivars, especially because we've
Mhm.
been doing that for our lion's mane project. We have many different cultivars of Heritium erinaceus and then also other Heritian species, and we're seeing big chemical differences between them. And also just looking at it grow, it's different. The
You can see
mycelium
the
grows
difference.
differently, different biomass, growth speeds, all of that can be optimized. And